Cytokines made in the early phases of an infection influence the functional differentiation of CD4 T cells. The differentiation of naive CD4 T cells into the two major classes of CD4 effector T cell occurs during the initial response of these cells to antigen in peripheral lymphoid tissues. This step, at which a naive CD4 T cell becomes either an armed Th1 cell or an armed Th2 cell, has a critical impact on the outcome of an adaptive immune response, determining whether it will be dominated by macrophage activation or by antibody production. The mechanisms that control this step in CD4 T cell differentiation are not yet fully defined. However, it is clear that it can be profoundly influenced by cytokines present during the initial proliferative phase of T cell activation. Experiments in vitro have shown that naive CD4 T cells initially stimulated in the presence of interleukin 12 and interferon gamma tend to develop into Th1 cells, in part because interferon gamma inhibits the proliferation of Th2 cells. As interleukin 12, produced by dendritic cells and macrophages, and interferon gamma, produced by NK cells and CD8 T cells, predominate in the early phase of the response to viruses and to some intracellular bacteria, such as Listeria species, CD4 T cell responses in these infections tend to be dominated by Th1 cells. By contrast, CD4 T cells activated in the presence of interleukin 4, especially when interleukin 6 is also present, tend to differentiate into Th2 cells. This is because interleukin 4 and interleukin 6 promote the differentiation of Th2 cells, and interleukin 4 or interleukin 10, either alone or in combination, can also inhibit the generation of Th1 cells. And that cytokines made in the early phases of an infection influence the functional differentiation of CD4 T cells. Have a great day.